we can have our next speaker present, who is Ekaterina Morozova, who is a postdoc at Brandeis University in the lab of Dr. Marder, and you can like start sharing your screen, and she will present like research on robustness and circuit dynamics of reciprocally inhibitory neurons to perturbation and modulation. And as a side note, I encourage everybody to visit Ekaterina's website with her amazing art inspired by neuroscience. And I'll post like the link in the chat soon. But yeah, please go ahead. Thank you.
Hi, everyone, and thank you, Davide, for such a nice introduction. And I want to thank all the organizers for giving me the opportunity to share my research with you. So I studied the dynamics of reciprocally inhibitory neurons called half-central oscillators. And actually, Krishna in his talk gave a very nice introduction of half-central oscillators and CPGs, which makes my work very much easier. But briefly, half-central oscillators consist typically of nonoscillotary neurons, but can produce oscillations in the absence of any rhythmic input when wired together. Reciprocal inhibition is a common circuit motif that underlies the production of rhythmic activity patterns. And it's essential for rhythmic pattern generation that underlie many motor patterns such as chewing, swimming, walking, and many other patterns of activity. So despite the apparent simplicity, these circuits are actually high-dimensional systems and can produce a variety of different outputs, including synchrony, antiphase spiking, antiphase bursting, and more complex patterns of activity. And in my work, I will be focusing on this alternating bursting pattern of activity called half-central oscillators. So our overarching goal is to gain insights into how neural circuits are able to produce reliable yet flexible output in order to adapt the activity to environmental needs and challenges. And to be more specific, I'm going to try to answer the question is how neural output is shaped by intrinsic properties and altered by modulators and external stimuli in perturbations. And one way is to understand the mechanism of how synaptic and intrinsic properties shape the output of the circuits is to have a control over some of the parameters that influence the output. So for these purposes, we use a dynamic clamp in order to create a reciprocally inhibitory circuit out of two motor neurons from the stemata gastric ganglion of the crab that you can see here on the picture. So briefly, the dynamic clamp is a technique in electrophysiology that creates a real-time interface between a computer and biological neurons in order to simulate nonlinear voltage-dependent currents and synaptic inputs in biological neurons. So it works by recording the membrane potential of a biological neuron with intracellular electrode, amplifying it, and passing it to the dynamic clamp system that contains a model equation of the desired current. Then current is formed and injected back into the neuron or into the other neuron, forming this artificial synapse. So at baseline, when pharmacologically isolated, the neurons that I'm recording from are just silent. And by injecting this reciprocal inhibitory currents and adding hyperpolarization activated current, I'm able to create this alternating bursting pattern of activity that you can see here on this picture. So previous theoretical studies have shown that there are two fundamental mechanisms of oscillations in reciprocal inhibitory circuits, which is escape and release. So escape mechanism is characterized by the inhibited cell depolarizing above the synaptic threshold, shown in red here, thus terminating the firing of the active cell. And the synaptic release is characterized by the depolarized cell falling below the synaptic threshold, thus allowing the suppressed cell rebound from inhibition. And by shifting the synaptic activation curve, it is possible to switch between the mechanisms. So we investigated the parameter space of synaptic current conductance and hyperpolarization activated current conductance for the reciprocally inhibitory circuits with different oscillatory mechanisms. and generated this kind of maps. So here in this maps, the color corresponds to the burst frequency, and the white space corresponds to the parameter sets for which no stable oscillations existed. So as you can see, the half-center oscillators exist for quite different and marginally overlapping sets of synaptic conductances for release and escape mechanisms. Further, for escape mechanism-dependent half-center oscillators, synaptic and H conductances have to be tightly correlated in order to create the robust bursting. And finally, the frequency is conserved along the different directions for two mechanisms because hyperpolarization activated H current produces an opposite effect on the frequency of oscillations and release and escape, decreasing the frequency of oscillations in release and increasing the frequency of oscillations and in escape. So likely in the real biological system, half-center oscillators depend on the mixture of the mechanisms. And further, it's also been shown that neuromodulators can shift the synaptic threshold, thus changing the mechanism of oscillations. So we were interested in how this maps look in the mixed case. So here you can see the evolution of the parameter space as we slide through the mechanisms by hyperpolarizing the synaptic threshold and going from release to escape. And you might think that operating at the mixture of mechanisms is beneficial because you can move either direction, but in practice, as you can see, half-center oscillators exist only for quite a small range of parameters. And this happens likely because biological cells, even of the same type, are never symmetrical with respect to the intrinsic and synaptic properties. And this leads to the fact that the mixture of mechanisms and the balance between the mechanisms is slightly different between the cells, causing the oscillations to be less robust. So now when we have some sense of how synaptic and intrinsic properties shape the output of the neural circuits, we want to study how they respond to the external perturbations. And one of the perturbations to consider is the temperature perturbation because it affects all biological processes to a smaller or greater degree. And neural circuits have to be robust to the temperature perturbations in order to ensure the survival of the animal. So dynamic clamp allows me to independently implement the temperature dependence in synaptic and hyperpolarization activated currents in order to study the individual contributions to the circuit output as a function of temperature. So I created a half-center oscillator in escape mechanism at 10 degrees Celsius and gradually ramped up the temperature. As the temperature being ramped up, the neurons hyperpolarized so that the top of the slow depolarization became closer to the synaptic threshold, shown in red here, thus switching the mechanism of oscillations from escape mechanism at 10 degrees Celsius to release mechanism at 20 degrees Celsius. And also you might notice that the frequency of oscillations is remarkably stable for a wide range of temperatures and only starts to increase as the mechanism of oscillation switches to the release mechanism. And if we were to further increase the frequency of oscillations, the half-center oscillator would break because the stable oscillations only exist when the synaptic threshold is within the the envelope of the slow oscillations. So this raises the question then how the neural circuits remain robust to the increases in temperature, especially if they initially operate at release mechanism. So it turns out there are multiple mechanisms that ensure the robustness of neural circuits, and one of them is neuromodulation. It's been previously shown that, that multiple peptides and neurotransmitters activate similar inward depolarizing current with a similar voltage dependence, shown here. And we used this voltage dependent and implemented this current with the dynamic clamp. I created a half-center oscillator with release mechanism at 10 degrees Celsius and ramped up the temperature to 20 degrees Celsius. As the temperature being ramped up, the oscillations attenuated and ceased to exist at 20 degrees Celsius. But the oscillations could be rescued by addition of neuromodulatory current or by simple depolarization, such as injection of the constant current. However, as you can see, the injection of constant current is less efficient than neuromodulatory current because the whole membrane potential is depolarized, leading to the less activation of the H current. And finally, it turns out there is an inbuilt mechanism that ensures the robustness to the temperature, which is the temperature dependence of the synaptic NH currents. So here I implemented the temperature dependence of the artificial currents with the Q10 of 2, meaning that the conductances increase 2 times as the temperature increases by 10 degrees Celsius. And as you can see, this rescued the oscillations and greatly increased the amplitude of oscillations, however, significantly decreased the frequency of oscillations in comparison to control, which is opposite to what happens in the real biological CPGs.
1 minute.
Thank you. And finally, by taking into account that all the activation rates of all the currents decrease with the temperature, we were able to account for this increase in the frequency of oscillations with the increase in temperature. So just to briefly summarize the effect of temperature on the half-century oscillators, increase in temperature differentially affects the frequency of oscillations in release and escape cases. And escape-dependent half-century oscillators are more robust to increase in temperature than release-dependent half-century oscillators. However, the oscillations could be rescued by the addition of neuromodulatory current. And with that, I would like to thank my lab. And of course, I would like to thank Yves Marder for being a very wonderful PI. And I would like to thank Peter Neustein, who is a technician in our lab and who was working on this project together with me and the whole lab for their support and helpful discussions. And this is the picture when we all were pretending to be crabs and one of our first Zoom lab meetings after the lockdown. And that's it. Thank you.
Thank you very much. It's the best thank you slide I've ever seen. Thank you very much, Ekaterina. And also thanks for the great presentation. If anybody has questions, feel free to post them in the Q&A button. So Brandon has one question, which is can you identify bifurcation between the escape and release activity? What kind of bifurcation is it?
Let me see, when you transition from yeah, it's a good question. I need to think about it kind of because the neurons they never stop oscillation, so they don't they rarely go through the transition between oscillating and silent states. So I don't know if it actually undergoes the bifurcation, but I do need to think about it. Yeah.
Yeah, we have also another question from Daniel. Do you think that robustness to temperature perturbations is still relevant in animals that haven't evolved homeostasis?
So I think it's mostly relevant to invertebrates, because especially crabs, because they actually come from Boston Harbor and they live in the ocean, and the temperature can easily change by that range of temperatures that I've done in the experiment. However, there were some experiments done, for example, heating or cooling the brain of zebrafinch, when they were observing also this changes in frequency of oscillations, which are kind of very similar to what we observe in invertebrates when we change the temperature. So I think the neurons are probably more sensitive to the temperature and more sensitive to the smaller variations of temperature. So they still have to be robust, but probably in a smaller smaller ranges of temperature variations.
Awesome. Okay, if there are no further questions.